Welcome to Thursday Dairy Update. This is Kathy Barrett with the Cornell ProDairy program, and the ProDairy program, along with Eastern Dairy Business, is pleased to be able to offer this webinar series. Today's webinar will be presented by Dr. Paula Ospina, veterinarian, PhD, with Quality Milk Promotion Services at the Cornell College of Veterinary Medicine. Dr. Ospina will discuss how to use information about bedding cultures to evaluate mastitis risk and how to use bulk tank information as an additional mastitis monitoring tool.
I have a lot of bulk tank information. The bedding information, which sometimes most people are most interested in, is still, in progress. We're just accumulating some of that data, but I think at least it gives us something to talk about, and hopefully I've left some room for questions at the end. So what I'm going to do is I have two topics to cover: the bulk tanks and bedding, and my focus again is how do we use that information and how do we go from there. So let's see if I can, there you go. So bulk tanks and bedding, looking at bulk tanks and additional tool for monitoring mastitis, and then I'll also be talking about bedding cultures and mastitis risk, which is research in progress at this point. Both of those actually are research in progress. And I'm also going to apologize, I am sick, so maybe you can hear that. Hopefully I will not cough throughout the presentation, but I apologize for my raspy voice. So to start off, we're going to talk about support and involvement. I just want to bring to your attention, some people may not be aware of this, but I think these programs are very helpful and give an opportunity both for research to evolve and then also for producers to get more information about their specific farm as part of this research involvement. So the New York Farm Viability Institute has funded both of these programs that I'm going to be presenting. The data that I'm bringing to you is coming from grants from the New York Farm Viability Institute. Two grants basically are the ones that I work with. One is the bulk tanks and their association with mastitis risk. So a lot of the information I'm going to bring to you about bulk tanks is coming from grants from this program. Excuse me, farms that are involved in what we call the new bulk tank program, which now costs $200 for a year. This grant offers $100 off of the enrollment, so it's only $100 for that year. We'll give you 6 bulk tanks and 6 reports, and I'll show you what all that information means and how that could be applied to your farm and your maybe decisions on mastitis, quality milk, and so on. The other is the bedding in the association of bedding with mastitis risk. And what we're looking for, the next phase of this project, so I'll be presenting the first phase, the preliminary information that we have right now. The next phase is we're going to be wanting to select farms from what we call the 4 different QMPS regions, which roughly speaking is northern New York, eastern, western, and then central here in the Cayuga area. We're looking for farms that are bedding with sand, sawdust, straw, manure solids, or paper. And then what we're going to do as part of this research section is we're going to go in and evaluate what's going in your bedding and how that's associated with mastitis, or at least we're going to try to do that, right? Because that's a lot of times what people ask. Do I have to sample my bedding? Is this an issue that is bedding related? I'm using manures. Is this still the right thing? Should I be doing something different? Can I blame the manure solids for my increase in coliform? Maybe. So, we're going to try to be answering some of those questions in the farms here in New York. There's definitely other programs, other research projects that go and look at some of this stuff on a national level, but some of the things that are specific to at least the eastern part of the country, northeastern part at least, very different from a California dairy, for example. So I think it's important to try to do some of this research in-house within the state so that we can get the information that directly applies to the dairy farms here. So how can you become more involved? If you'd like to become part of the bulk tank program, there's a brochure. If you follow the link, here, and I think these slides will be available. You can just click on the link and it'll lead you to the brochure. And you can send that to me, and I have my email all over this presentation. You can send that filled out brochure or information to me, and then usually what you have to do is just mail it in. The brochure prints out with all the information that you need. Once you become part of that bulk tank program, you will get the reports from your participating regional QMPS lab. If you, and here's my email, if you wanted to be part of the next phase of the bedding project, just email me with this information. I need your name, your email and phone number, and let me know which one you prefer to be contacted by, or with number of cows you're milking, your address so we know what region you fall under, what type of bedding you use with your fresh cows, what you use with your dry cows, and then if you have different bedding elsewhere. Like some people will have deep mattresses here, or deep beds here, mattresses there, etc. So just give us a little bit of a description of what you're doing for bedding, and then that way we can see how you fit into the project and see if we can get you in. And again, what we would do is we would go in and sample your bedding, sample your cows, do a bulk tank at the same time, and try to tie that, that question that we all have: how is bedding and mastitis risk associated? So just wanted to give you a little bit of background of how this all works and what might be coming next. And lastly, with the bulk tank program, we may be restructuring some of that project. And this, in addition to receiving your bulk tank reports, you might be eligible to receive some training associated with what we find on your bulk tank. We're working with New York Farm Viability right now to see if we can make that happen. And that's a great opportunity, because if we find high counts or low counts, and we want to just make sure everybody's doing the right thing, you have the opportunity of having, I'll be going out, I have a student coming in that's going to be doing her master's on this, coming out to do training in Spanish, so that opportunity is there, but you have to be part of the bulk tank program to potentially be eligible for the second part. We're not even sure if we can get that to happen, but that's our goal. So just wanted to give you some updates on that, so you have the opportunity to participate if it's something that interests you. And again, feel free to send me email with questions. I'll be happy to try to elaborate a little bit more if you have questions. Okay, so now to the meat of the presentation. We'll start off with some information that we gather from the bulk tanks. So when you submit a sample of milk from a bulk tank, you'll get a report. There's different types of reports that you get, but from the bulk tank project, this is what we'll send you. So you'll have, where the sample came from, bulk tank, etc. And then these are the test results that we review. So we'll give you detected versus not detected of environmental and other pathogens. We also look at the contagious pathogens, Mycoplasma, Strep Ag, Staph aureus. And then the new part of this bulk tank program, if any of you guys are familiar with the old bulk tank program, is that we also give some quantitative counts. So we look at how many total streps there were, how many total staph species there were, so strep species, staph species, and total coliforms. So these numbers were new to this program, and that's part of what ended up kind of getting that grant process started, because we wanted to know, are these numbers, do they mean anything? Are they helpful to anybody? And what about this qualitative yes or no information? Is this worth anything? There's always lots of people looking at bulk tanks when you send your milk in and so forth, but can we look at this from a mastitis, risk of mastitis point of view? So we have this information. Okay, now what? Ideally, lots of people will tell you that benchmarks are nice because you have at least some point in the universe for reference, but they're difficult because you don't necessarily want to compare to a different farm that's doing something differently and may not be, their numbers may not be really related to what you can do on your farm or what they can do on their farm, etc. So we always recommend establishing a baseline within your farm and comparing your numbers to yourself. You know what you can do, you know what you can accomplish, you know what happened when things went wrong, and that can help you get that information. So with the bulk tank program, for example, after you've had several bulk tanks, you'll get information that looks like this. So we coordinate with the haulers and collect your milk sample. You don't have to send it in. We figure it out, and then you get a report that says, this is what we found in your bulk tank. These were the counts, those quantitative numbers that I showed you, and these are the bacteria or pathogens that were identified in that sample. And then we always highlight Mycoplasma just because sometimes, that can pop up as a positive and be a surprise to some people. So in this farm, we're milking about 650 cows, and we're going along, if we look at our strep species here, more or less okay. Maybe, again, we don't know if this is a good number or bad number yet. I'll show you some of that in a second. But then all of a sudden we have a change, right? So regardless of whether this 120 is good or bad, we know that 120 is very different from 2,000. And so, what does this mean to this farm, and can we do anything to change it? You might also notice that on this farm, we have Staph aureus pop up, a couple of tests, but not every single test. And what does that mean? We know from individual cow data that cows can shed intermittently, so it's not surprising that we'll see it pop up or not pop up in the tank, especially with 650 cows. You may not find that. But if this is a herd, for example, that doesn't think that they have Staph aureus or think that they have Staph aureus controlled in such a way, then they shouldn't be seeing this. This is a good alarm bell. We've also had a couple farms pop up positive on mycoplasma on their bulk tank that, didn't know that they were dealing with any mycoplasma. One very, we talk about success stories sometimes when we're dealing with the programs, and this farm in particular, for some reason, was under the impression that fresh cows were getting cultured aerobic culture and also getting a mycoplasma culture. And at some point in time, that was taken off. So the fresh cows were not being cultured for mycoplasma. All of a sudden, the bulk tank tested positive for mycoplasma, and this alerted them to an outbreak of mycoplasma that was starting. So this and other information, obviously somatic cell count increasing, sick cows, things like that, all came together. And even though this is not a way to monitor for mycoplasma, it's another safety net to, to keep that in mind. If you're really concerned, string sampling is a better way to, to check and find these mycoplasma samples, because there's concern about when you pull, you might not be able to find it, and so on. So again, not advocating that this is the way you should check and rely on this, but it is another safety net. So other farms, for example, here we go. We're going along. Again, not sure yet if these numbers are good or bad, but we know that these numbers are very different from this 22,000, right? So we don't have to know this is good. We assume that this is not good, being so high. And what about this coliform numbers here? 80, not detected, and then all of a sudden 9,000, 6,800, and 1,000. And here we are, mycoplasma positive. So this is that farm where there was just lots of stuff going on. All of a sudden they popped up mycoplasma positive. This is actually not that farm that thought that they were testing. This is a farm that knew that they'd had mycoplasma in the past and had it under control, and then all of a sudden, not under control. So lots of things going on there, and the bulk tank program helped them identify some of these issues. This is the somatic cell count for that farm. This one here with these numbers, so we can see in October 2015 we spiked with coliform counts, and in January we spiked with strep species counts. So October and January, so here we have the spike. We don't have the data from back here, but we do have the data here. So we're not only looking at, sometimes these coliform bacteria counts are associated with cleaning problems, not enough hot water, the detergents aren't, set up correctly, somebody forgot to buy it, or, all those things happen. But sometimes we do see this in addition to potential problems with cleaning. We also see this as a result of cases of mastitis. So we had an increase in somatic cell count, lots of stuff going on in this farm. Again, not the only thing that was being looked at, but it definitely helped to monitor this. And again, when this farm is now using its own self as a benchmark, it can easily see, here we were going along, all of a sudden this is not good. We're getting back down, we're getting better. Hopefully we'll be on the mend. And, as far as somatic cell count, lots of things have been done to help improve this, and we're seeing a little bit of evidence of that. So here's another farm. This was the first bulk tank that was received from this farm, September 2014. And again, without knowing what the benchmarks are, what's a good number, what's a bad number, we can see that this is probably not a good thing to have these really high numbers, especially when, just briefly, I've shown you some that say not detected, or 80. So when we have 33,000, 32,000, it makes us concerned that maybe this is not a good thing. So these numbers seem high, but how do they compare? This is a farm milking 500 cows. This is their somatic cell count. So, when we got this bulk tank sample, we were up here. So definitely lots of stuff going on there. This is skipping away from that farm, but just giving you now the benchmarks, which sometimes, again, are a good thing because you have at least a point in space to compare, especially when you have your first bulk tank analysis or any other measure where you don't have historical data within your farm to compare to. So what we have here are bacteria counts in the bulk tanks, and these are the median numbers. So when we looked at this distribution, it wasn't a normal distribution, which is that nice bell curve that we sometimes see. It was shifted. So we did a median, which means that half of the farms that submitted samples, or it'd probably be better to say half of the tanks that we've evaluated, because some, farms have submitted several tanks. Half of those tanks were below this, and half of them were above. So here we have a coliform of 20 staph species, of 60 and strep. So half the farms have 60 staph species, half of them more, half less than 20 for coliform, half of them more. And remember that farm I showed you had about 33,000 coliform, about 800 staph, and about 33,000 strep. So, they were way on the, on these ends of the scale when we were examining, comparing them to other farms. And again, the best thing to do is to compare within farm, but when you don't have that opportunity, these benchmarks are give you that that opportunity to do that. So this farm, elected to get some extra help. Obviously, things were going on that were concerned. So evaluation of the cleaning system was important, especially with these high coliform counts. And water temperature was a problem there. And then there were new cases of mastitis, strep-based, that we went over reviewing how to identify mastitis, how to treat mastitis, and so on, so that we could pull these cows out of the tank and treat them appropriately. So just as a quick review, for the pathogens that are reported under the count, the ones that we give you counts for, coliforms generally just are talking about a gram-negative environmental pathogen. So it's not necessarily, it's an umbrella term for your gram-negative pathogen. So it's not E. coli specifically, but it can contain E. coli. It's not Klebsiella, but it can contain Klebsiella. It comes from manure solids. It can come from soil, water, and bedding. Clinical mastitis will vary depending on which pathogen it is. Klebsiella, for example, E. coli, for example. You can have several clinical infections during the lactation. These guys may, depending on how severe it is, may require supportive therapy, IV fluids, and it can also be associated with cleaning issues, which is what I mentioned already. So keeping cows clean and dry will help eliminate a lot of this. Cleaning stalls frequently, because we do see it, in manure. Coliform vaccination sometimes will help should help prevent the clinical mastitis severity. Remember, though, it's not going to prevent mastitis. It's going to limit the severity of it. Adequate bedding, and we'll talk a little bit of bedding later. Overcrowding can be an increase the risk factor. Muddy, wet, standing water, any of those things, maintaining milking equipment, and then again, coliform counts greater than 1,000 are, we're worried about dirty milking equipment. When we look at strep species, again, we're at an environmental pathogen. Similar details as what we find there, but we see this more in fresh cows. Excuse me. Again, keeping cows clean and dry. They can respond well to an intramammary treatment during lactation, and can also sometimes be associated with cleaning issues. When we look at Staphylococcus species, and now this is different than Staph aureus, which is a contagious pathogen. We're looking at the environmental forms of this. So this is an environmental pathogen, similar findings. We can see an increase in somatic cell count. Infected between milkings, but can be spread during milking, dirty teeth and udders. Bedding management is important, especially over the dry period. May not respond well to intramammary treatment during lactation, but some cows will respond well to dry treatment to get rid of some of these staph species that can plague a herd. So that first farm that I showed you, or the farm that the first count that started off in the thousands, this is some of their later bulk tank data. So April 2015 until February, so we're currently now. You can see, you know, sometimes we get things that creep up here and there, but much better control of what is going on with these with the bacterial counts, and then also the species that we're finding in the bulk tank are also kind of more routine, so no contagious pathogens and so on. Continuing to just establish those baselines or potential benchmarks. What we have right now is just descriptive information, which is the first step. The next step is then say, okay, we have these farms with these tanks that are positive for this. What makes farms that are high similar to each other, and what makes farms that are low similar to each other? So this is the next phase of the project that we're in right now. We're evaluating what those risk factors are that would make a farm, you know, have a lot, have high counts, more likely to have contagious pathogens, etc. So right now, the basic information I have to share with you guys is just based on herd size, and this is pulled directly out of a Hordes article, and I can give you that link if you're interested. Just email me. That goes through and you know, just establishes bench point for that. So what we see here is the herd size, just because we have to divide the data somehow, and said how many tanks were positive for example, Mycoplasma, Staph aureus, or Strep ag, so our contagious pathogens. And we see that Mycoplasma we saw kind of throughout. We had, you know, a few tanks that from big herds, and again, there is a dilution factor here, so it doesn't mean that big herds don't have Mycoplasma, it just we might be able to find it in a tank. So we see that happening there. Staph aureus, we see it all the way through. Strep ag, very few cases, and only one part really that was positive here for Strep ag. So that says a lot about the control of this organism. Contagious pathogens continued. Don't forget, management of contagious pathogens is not novel, but don't forget the essentials. You need proper milking techniques. Milkers have to wear gloves. Pre and post dip is helpful to help eliminate. You know, after that milk unit comes off, if they had a contagious pathogen on it, and the cow, it's on the skin of the cow, you put that post dip on. You can limit the risk of that cow getting that mastitis from that pathogen. Clear identification of contagious animals. So this seems a little bit basic, but many times if you collect all these Staph aureus cows in a pen, but don't put leg bands on, if one animal gets out in the wrong pen, you know, now you have this animal floating around and being a potential source of infection for other animals. We want to milk them last so that we go through a wash cycle before we put, you know, go back to the rest of the herd. And even though this seems, again, very basic, I've been on farms where, yeah, we milk them last, but for different reasons, different scheduling, the wash cycle has been changed. So we can milk them last in the group, but the wash cycle is not done until the next group is through. So again, we've forgotten the point of managing these contagious pathogens. Don't forget that you need that disinfectant and washing equipment between these animals and other animals. In our contagious pathogens are Staph aureus, Strep ag, and Mycoplasma, but we're starting to see a little bit of evidence of other pathogens popping up here and there. The pathogens that we normally consider environmental pathogens, which we see as pathogens that are all over, maybe in the manure, just in the environment, free living, that can then infect the cows between milking. Sometimes we're seeing these pathogens act as a contagious pathogen. This next slide, I'm going to jump ahead and I'll come back to these slides. So this next slide here talks about these other pathogens that are normally not considered contagious pathogens but are behaving that way. So here are some gel bands from looking at DNA sequencing in A through H. So A through H here, right? These guys are DNA sequencing done in May of 2015 on a farm. And then I through Q were done here in 2016. And what I want to highlight is these bands that are in purple, on my screen at least, you know, look very similar. And what that tells us is that these pathogens are related to each other. And if you think about the life cycle of a bacteria, which is about 20 minutes, and then think about how long it's been since May 2015 until January 2016 in 20-minute increments, the generations between these bacteria are in the billions. And here we are having very related bacteria. So a lot of times we would think, oh, this might be Staph aureus, or something like that that can be contagious. But this is actually Klebsiella on a farm. And so we're dealing with a contagious Klebsiella pathogen. So what do we do about this? We need to treat this as a contagious pathogen. So we're working with this farm, trying to set up a pen that just groups all these cows together, just like you would do a Staph aureus pen, identify these cows, make appropriate culling decisions. If these guys don't cure over the dry period, so that's the question that we have right now, they should, but we're going to be testing when they freshen and so forth to make sure this Klebsiella is out of their system. So let me go back to those slides that I skipped over really quick. I don't want to spend too much time going over stuff that you guys are aware of, but mycoplasma, contagious pathogen, difficult, impossible to treat, difficult to manage until you identify the cows and at least try to cull or group them in a pen. But sting samples is probably the best way to do it if you're concerned about it, an outbreak or just identifying the mycoplasma animals in your herd. Or some people will submit samples, fresh cows, and check for mycoplasma at that time. Staph aureus, I think you guys are all familiar with, contagious organism, lives in and around the udder, spread from cow to cow. So we need that milk contact between cows in order to transmit this disease. Strep ag, like I showed you in that graph, we don't see very much of anymore. So it's a contagious pathogen. All the same things apply, but we're just not seeing as much of it as we used to. So this is good. We have to dig for this slide actually. So again, don't forget other pathogens. Excuse me. Other pathogens can also behave as a contagious pathogen. So be aware of that, and if it looks like the things that you normally do aren't working, get some help. We can get in there and do some more testing to try to figure out some of this behind the scenes stuff going on. So gram-negative pathogens, as far as the bulk tanks are concerned, here again we have the number of tanks that of 250 tanks with fewer than 250 cows, and we had E. coli in all of them, Klebsiella in all of them, Pseudomonas in all of them. Not a lot of Serratia, so that's good. But we did see E. coli and Klebsiella kind of our two prominent gram-negative organisms that we worry about. Quick review: E. coli is our environmental pathogen, gram-negative. It's a manure. Same thing that we saw for the coliform slide: keep cows clean and dry, reduce overcrowding, appropriate and adequate bedding, etc. Klebsiella, our environmental pathogen. We can get some chronic cows with here. So what's tricky about this Klebsiella, and then just relating back again to this farm where we have this contagious form of Klebsiella, is that we can have chronic cows that are maybe subclinical, or we see in this farm at least that we're seeing some subclinical Klebsiella. So these guys are, can have a high cell count, but don't have a clinical mastitis, no redness, no pain, no abnormal milk, and they're shedding this pathogen and infecting other cows. Pseudomonas, an environmental pathogen. We see this more in the water. So it's coming from contaminated water supplies mostly. It can affect the bedding in addition. Teacups can spread this really nice. You get some chronic infections, and this can be associated with a high PI count. Other organisms that we look for in the bulk tank: Prototheca, T. pyogenes, and yeast. We don't see a ton of Prototheca, but we do see it in some big herds. We're dealing with a couple of herds milking over 2,000 cows that are having Prototheca issues. T. pyogenes, we see that here, and again, getting a 0 on these large herds doesn't mean they don't have it, just we might not have been able to pick it up, and we see yeast throughout the different herd sizes. So just as a review, Prototheca is an algae found in the cow environment, water, soil, bedding, and so forth. Early detection of the infection is key. There can be some cow-to-cow transmission, so it can act somewhat as a contagious pathogen. we can have very high somatic cell counts with this, and the infections can last through lactation. It can be the lifetime of a cow. You may have to dry off a quarter. You know, you definitely want to be able to identify this if you have it. So when we find them in these bulk tanks, you know, like we found them here and they pop up, you know, we talk to this farm and we're aggressive about trying to figure out what changed or what happened. Some of these farms, we know they have a residual problem because of water things that they had issues with in the past. But other farms, you know, if they pop up on a farm we don't know it's a quote-unquote Prototheca farm, you know, we're talking to them quickly. So this Prototheca herd, when we had the spike in somatic cell count here, we had Prototheca club identified in the bulk tank, and we also had lots of other stuff going on. You can see the coliforms here, 17,000 in September, which is when, you know, when we were getting these spikes up here. So again, keeping track of some of these counts can help us identify some of the other problems that are going on, and at least, you know, alert us something's going on so that we can go in and investigate a little bit more. T. pyogenes, sometimes also considered summer mastitis. We see it with damaged tendons. Treatment outcomes are poor, so we want to make sure that you guys are evaluating your equipment, checking tendons if you don't, to make sure that we're not damaging those tendons and try to minimize some of the cases that we see here. Loss of a quarter is not uncommon. Excuse me. And sometimes we'll see this pop up positive if the milking unit is attached to a blind or dried off quarter. So sometimes we'll see positive pop up on the bulk tank. We go in and do some more investigative work and not find it so much, but sometimes we can see that, you know, if this mistake happens. Yeast is found all over the place. Whenever you look for it, you can find it. But when we find it in a cow's udder, we see it because we've introduced the contamination mostly, either through medication, even with dry cow, or treating for mastitis. If you see mastitis and you put antibiotics in this cow, if you don't follow very sterile procedures, you can actually infect the cow with yeast and give her yeast mastitis. Or if she originally had a yeast mastitis and now you're adding antibiotics to that udder or that quarter, you can make the yeast mastitis worse because now you've gotten rid of any potentially good bacteria and left the yeast to overgrow. Okay, so that was bulk tanks, but basically, just in summary, you know, we are using this information, and the next step again is then to go and look at the data that evaluates why some of these herds, you know, will have these contagious pathogens, or I mean, everybody has a little bit of it, but you know, what the distribution is and if there are any risk factors that can be associated with that. And again, hopefully we'll be able to provide some training which will help us identify more of those risk factors as we get on these herds and actually talk to the milkers and evaluate their system as they go through. All right, so bedding, lots of options. You know, what do you pick? What's the best option? And the reality is that there's no right answer for everybody. There's no one answer that says, you know, this is the best answer. Sometimes it's a cost issue. Sometimes it's a facilities issue. Some facilities can't handle sand. They might want sand, but they can't handle it. But the ideal situation is we're giving that cow the opportunity to lay down in clean, soft, nice, comfortable bedding, so that when that udder hits that bedding, you're not introducing any contamination to that udder and challenging her immune system to fight off infection. Obviously, this is difficult because these guys are tracking in stuff from the alleyway, so whatever we can do to keep those alleys clean and so on, and whatever we can do to maximize the appropriate use of stalls, right? So if every herd looked like this, I think lots of problems would be solved, right? We have appropriate use of these stalls. If this cow were to defecate or urinate, it's going to come off. It's not going to affect what's going on in the udder. You can see how soft and fluffy everything is. I mean, I don't know if you can get a better picture of this, and I don't know if I can. I actually stole this from somebody else. I've never, you know, had such a nice picture that I could capture in that moment. This is ideally what we want. That clean, soft bedding that doesn't challenge the cow's immune system. So to start off with, bedding samples, lots of people have questions about these because we don't know what it means when we get some of these results. Sometimes we see that there's lots of variation in how people collect the bedding samples, when in the day they collect bedding samples, how do we send it into the lab. So we'll have some variability in the results that come out of here or any of your QMPS labs that run the bedding samples, depending on how they're packaged, when they're collected, etc. So the project that we're working on with New York Farm Viability, as I mentioned previously, has two phases. The first phase is answering some of these questions. When do we collect it in the day? How do we collect it? How do we send it? How many samples do we collect? The next phase of this, which is, what I'll encourage you guys, if you're interested in what's going on with your bedding, to sign up for that second part, see if we can get you coordinated to be on the program or on the project. Basically, it's them saying, okay, we figured out how to collect these samples and what we want to do. So now we want to see what goes on farms and how that changes mastitis risk and how that changes what we see in the bedding. So we looked at 3 farms that were using digested manure solids to bed fresh cows. And the reason we focused on fresh cows was because these guys are at the highest risk for mastitis. And so we wanted to, take advantage of the time and budget that we had and focus on the most variable bedding substrate that we could find. So manure solids seems to, for us at least, seem to be the most variable in a lot of aspects: moisture, bacterial, pathogen count, etc. And then fresh cows, again, because of all of the different things that are going on with them metabolically and so on, would also give us the most variability. So we looked at 3 farms, and what we wanted to identify was how to sample stalls and then also how many stalls to sample. If we were going to sample the fresh pen, how many stalls did we have to sample to get an idea of what was going on in that pen? So the 3 farms we picked were Southland Cow Dairy with 100 stalls in the fresh pen. One of the things that we did see that made a big difference in the bedding samples that were collected was whether the farms used lime. It either mixed it in with the bedding prior to applying the bedding on the stalls, or mixed it on the stall, just dusting over the end. So we did see that as a big difference there. But we had different size farms and different size fresh cow pens, obviously. So moisture. It's hard to do it in this format, but think to yourself, which would be more moist, fresh, or used manure solids. So I'll give you a second to think about what would be more moist. And you might have some opinion based on personal experience or just other information. But based on just these 3 farms, what we found, here's the moisture in fresh bedding. So we have our 3 farms here. You can see there's a lot of variability even between these 3 farms. So again, once we start evaluating other farms, we might get a little bit of a bell curve here, but this is what we have so far for fresh bedding. And then used bedding, here we have that same distribution as we saw with the fresh bedding, but it's a little bit higher. So, and then we think that sometimes they dry out, and potentially this might change seasonally. So if we had fans running, the used bedding might actually be drier than the fresh bedding, but this was during the winter season, so this may change seasonally. But this is what we're seeing with these 3 farms so far, and again with manure solids. So what did we see as far as bacteria count in these 3 farms? Basically, the farms that were using lime, their fresh bedding didn't grow any coliforms. We still saw some strep and some staph species in these farms in the bedding that grew, but the farm not using lime was the only one that grew some coliforms. When we looked at used bedding, everybody grew everything. We saw coliform in all of them, strep and staph and so forth. And again, because we only have 3 farms, it's hard to do statistics to say whether these are statistically different than the other. All we can say is they all grew coliform, they all grew staph, they all grew strep. Maybe this farm grew a little bit less staph, and maybe this farm grew a little bit more strep. But it's hard to really make any more statements based on this just very descriptive data. The thing that we started to do with these, even with the small sample of 3 farms, is that we wanted to start to evaluate that association between what we saw in the bedding and what we saw on the teat ends, imagining that, you know, this cow goes and lays down in that stall, and whatever's in that bedding probably will end up in that teat end, and if it's on that teat end, maybe it will end up as mastitis in the future. You know, that's kind of the idea, right? We're trying to identify the association between bedding and mastitis. So what we did was we swapped teat ends, and then we cultured those teat ends on different media, selected media, so that we could see if there was a lot of streps, if there was a lot of staph species, if there was a lot of coliform species. And then what we did was we did a semi-quantitative result on this. So if the cultures had a bunch of streps, you know, we call them high. If they had, and this is very subjective, but this is the same lab that did this, so we can compare these between each other. If they had a little, you know, medium amount, they got the medium. If they had low amount, they had low. And then if they had some negative plates, we had a negative. So what this farm, for example, says, they had fewer numbers of plates with high amounts of streps, you know, more with medium amount of streps, and then a lot more with low amount of streps. So, and this is the kind of the same path here, but we see a lot more low streps on farm 2. And then in farm 3, we see a little bit of a reverse relationship between the streps and the between the high and the medium here. So, you know, it's scaled like this on these 2 farms, and then not so much here. So again, this is where that next step of the project just goes in to evaluate, you know, what's different on this farm that we see more of these high versus this in the relationship to each other, and also just in relationship to other farms. You know, why are we seeing almost 30% of the teat ends coming back high in here when we're down in the 10 to, you know, almost 20% here? You know, what makes this farm different? And we don't have that information yet. That's our next step. When we look at staff, we had the fewest number of high growth, then medium, and then low growth, and we had a lot of medium growth here, and then equal medium to low growth, and so forth. So again, though, the lowest number of plates were the ones with high growth, and then medium, and then potentially equal or lower of the low growth plates. And this was looking at staph species. Then when we looked at coliform species, a lot of the farms had low growth when we swabbed their teat ends. And then farm 1, you know, very predictable and kind of persistent as far as their patterns. We had, you know, fewer of plates with high growth, a little bit more moderate growth, and a lot more of low growth. So, you know, most of the plates that we saw had low growth. More here and more here, and then even some negatives here in farm 2. So this is looking at coliforms. One of the things that's difficult to evaluate when we look at coliforms is that this might actually be a reflection more of our sampling technique. Coliforms need a little bit of moisture to be happy. So if they're on the teat end, they might be right around that orifice. And we were trying to avoid getting milk on our teat end swabs. So we went as close as we could without getting any milk on them. So we might have missed some of those coliforms that are on those teat ends that we worried about with manure solids because we didn't get close enough to that teat end orifice. But obviously we did get some growth. Just a lot of the plates had low growth. If we had gotten closer, maybe we would have more plates with medium or high growth. Okay, so how do we collect these bedding samples? Be systematic, always at the same time. And what that means is when you go to the stall, approach it in the same way every time so that you can compare your bedding samples between each other. It's really hard to compare to other farms because there's so many variables, and even within farms, from this very, you know, small study that we've started, we saw a lot of variability between stalls. So it really depends. You really want to be systematic and collect the stalls in the same way. And I'll show you what we did and what we're recommending. Always at the same time. What I mean by this is we elected to pick the dirtiest bedding. So we wanted to collect bedding before new bedding was applied. So the dirtiest it could be before we had new bedding. That can be difficult on some farms because by the time it's due for new bedding, there might be no bedding on the if you have mattresses, for example, right? So you have to pick what works best for your environment and what you're trying to accomplish. Obviously, we want some bedding so that we can send it to the lab and have enough of the substrate to do the test that we need to do. But we collected before new bedding was applied, and we collected it after it was raked. And the reason we chose that was because I know that if I go and I put my hand in manure and put it in a bag and send it to the lab, they're going to grow a lot of bugs. You know, that's not a surprise to anybody. But what I want to know is, even though, yes, some cows will come back to that stall, and if it's dirty, they will lay down in that manure, and that's fine. Some cows will be exposed to that environment. I want to know what's in the bedding, and hopefully not all of the stalls are like this, right? Hopefully they are clean, especially after raking. So what we did was, again, we were focusing on the fresh cow pen. So when the cows left to go get milked and the guy was walking through the stalls raking, right after he raked, we went in and collected our sample. And even though I don't have this data to share with you today, the other thing that we did was that we scored our stalls and said, before he raked, how much of the stall was covered in manure. And right now we're in the middle of evaluating that data. The number of stalls, usage of stalls, things like that, does that affect what we saw as the outcome on the teat ends, in the bedding? You know, the farms with more dirty stalls before raking, do they have higher counts and so forth? So again, it may come down to just very simple things that we already know, right? Cleaning, providing fresh bedding, all those things are very important to minimize the risk of mastitis. So we focus on the end of the stall, right? This is where the udder comes in contact with the stall. This is where we want to focus. And obviously these stalls don't have any bedding on them, but, you know, just to give you an idea of where we are in the stall, we used like the 2-gallon Ziploc bags, and then we changed gloves between the stalls because we collected like 40 stalls per farm and so forth. I'll talk about how many stalls we think you should collect, and again, we're working on those numbers right now, but at least we have a start. So we don't want to contaminate what we find in one stall to the other because there is some variability even between stalls. So we want to focus on the area that the udder touches. And what we've done, there's 2 methods, I think, of collecting. One is you divide this back end of the stall into a grid of 9, and you kind of do, you know, in vet medicine, we'd call these punch biopsies, where you stick your hand and grab, you know, a handful here, and maybe you're very systematic and you do, you know, a handful here, and you split this in 9 squares, and you do 1, 2, 3, 1, 2, 3, and then you mix it all in the bag. What we ended up doing was saying, you know, we really want the surface area. And so we ended up scraping that top layer, right, into the bag, and just scrape the top layer, because we said, and we weren't scraping down deep, especially in deep beds. We found that if we dug down deep, we found lots of coliforms. If we collected from the top, we found lots of strep. So there was a difference in bacteria, what was on the surface of the bedding versus what was deep in the bedding. And we wanted to know what the cow was coming in contact with. So when she came back and laid down, and her udder hit this stall, you know, that top 1 to 2 inches is what that udder should be touching. So we just scraped that off and put it in the bag. You know, always at the same time, we recommend after scraping, just again, because manure is going to change your results drastically. And even though it is true that if there's manure in the stall and that's what they come in contact with, that's going to cause, you know, that could be bacteria and cause added risk. But we might be able to appreciate that information by scoring the stall, not by sending in samples of manure. Because if you sample manure, you'll get high numbers of bacteria. You want to focus on sampling when the cow's udder comes in contact with, and then especially after milking, when we consider her to be the most vulnerable coming back, her teat ends might still be open, etc. So sample, you know, the back end of the stall, get the Ziploc bag, use the gloves, be systematic. We recommend scraping the surface of that back end, avoiding manure. So if they've come in and scraped out the manure, so hopefully it pulls the bedding back. This is what the cow is going to come in contact with when she comes back from the parlor. So this is what we want to know, we want to know what's there for her to be exposed with. You could argue that before she left to go into milking, that's also a high-risk time. But again, because there's manure in the stall and we're trying to avoid the manure, that gets a little complicated. So I think you just have to decide what you're going to do and just be consistent with it. So how do you send the samples in? We evaluated frozen samples versus fresh samples, and we didn't find a difference in counts. So if you have to send us samples and you can't get it to us the same day, freeze it. Because bacteria will grow very quickly in warm weather, and bedding material is great at growing bacteria. So we don't want to affect those counts by having a sample that's been left out in warm temperature. This is a bigger deal in the summertime, but we want just consistent temperature. You know, if you can get it into us that same day because you live close, you know, you're situated near a lab that can process this and you can drop it off in a couple hours, you can probably keep it cool, put it in a cooler with some ice packs. But if it's going to be mailed to us or anything like that, you want to stick it in the freezer, freeze it, and send it frozen. How many stalls to sample? We're working on this number because, again, what we did in those 3 farms, we sampled all the stalls in the pen, or as many as we could based on our budget and time and so forth. And so we're looking that you probably need at least 5 stalls, and that number may change as we look at some more of that variability that we were finding. We have one more farm to sample, and we'll finalize these numbers. And then the big thing is that there's a debate about, you know, whether we want to look at just stalls or whether, if we're evaluating bedding, you know, are we evaluating the environment? And so this might be part of an environmental exam is what I would think about it. So sometimes we're looking at feed samples, sometimes we're looking at alley samples, sometimes we're looking at manure samples, because we want to identify, you know, other sources of these infections. For example, I go back to that Klebsiella farm that, you know, we're working with. We're going to go and collect feed samples, alley samples, and manure samples, even though we are suspicious that this is all, you know, contagious pathogen coming from other cows chronically, subclinically infected with Klebsiella. But we want to just make sure that, you know, it would be very unlikely that we would have that same bacteria that's so related to each other, you know, in the environment, and the cows happen to be infected by it. But we want to look at this as an environmental exam, and bedding is just part of it. So in summary, just want to remind you guys that bulk tanks can be used to help monitor mastitis pathogens, looking at changes in bacterial counts, appearance of contagious pathogens. The bulk tank report helps keep track of these changes, and we've had lots of farms use these as part of their vet meetings, you know, to go over some of this stuff. Sometimes the parlor gets forgotten a little bit, so this kind of helps remind people of what's going on in there. Bedding evaluation, our research is still early and ongoing, but we want to evaluate the risk of mastitis given bedding pathogen load, and, you know, I'll ask you for your help on that if you guys want to sign up for that second phase of the project. You know, this gives us opportunities to learn more about your farm. So if you want to sign up for the bulk tank program or for the bedding project, you can contact me. I want to give thanks to the New York Farm Viability for funding the research opportunities, and for you guys that let us explore these questions on your farm. And as a new parent, I have a little 1-year-old that's going to turn 1 in a couple of weeks. So of course I have to include the picture. This was us at Christmas, and here's my email if you have any questions or, you know, want to get involved with any of the programs or projects. Again, I think I probably have more questions about bedding than answers. You know, that's evolving. But I think the bulk tank project, we definitely are seeing some good information coming out of that. And then, you know, going on to the next phase where we might be able to do some on-farm training in response to some of the changes that we see in the bulk tank. I think there's lots of opportunity there. And then I hope that this information was useful if you guys are looking at monitoring some of these things using some of the reports that we can offer. And that's it for me. Thank you.
Oh, Paula, thank you very, very much. This was excellent, and your little one is adorable, really cute. So we have a couple of minutes. So, folks, if you have questions, go ahead and put them in the chat box, and we'll get them answered. While folks are doing that, I had a couple of questions, Paula. The first is, outside of your study, the bulk tank and the bedding sample, is there an opportunity for farms to be able to do either of those? And how the bulk tank, do you see a lot of folks doing samples on a regular basis and that sort of thing?
Yep. So thank you for that. That's a great question. And basically, the bulk tank program exists outside of my study. It's an own independent thing. It does its own thing. my study just gives you money so you can do it cheaper. And then I use that information to help educate, farmers so that when they're receiving these reports, they know what to do with them. So the bulk tank program exists and it will continue to exist past my study. The difference is right now we have money to help subsidize that cost. The bulk tank program costs $200 for 1 year, and the grant that we have through New York Farm Viability gives you $100 towards that, so it only costs $100 for that year. The bedding, you can always submit bedding to QMPS. You have to call the lab because not every lab is set up to do it right now, and it depends on it's very intense process. Vacation or whatever, we might not be able to do it in Ithaca, but Skill might be able to do it. So we will just ask you to, send your samples there or here, depending on what's going on. But you can always send bedding samples outside of this project. We just use these projects to help us analyze this information so that when you get your report, we can help you understand what's going on. And also with bedding, the question is always, how do we sample? Where do we send it? How do we send it? So we're hoping, to fill in some of this information with these research projects.
Great, thank you, Paula. And then I had just another question. So and this would be on the bedding project. So, you said you did teat-end swabs, and you probably said it and I didn't catch it, but so you do these swabs before any milking procedures or just before you put the machine on? And the reason I'm asking is, I'm wondering as this project goes forward, how you correlate the actual cows that have infections versus the teat swabs and the bedding, and then I suppose down the road the bulk tank. So could you just clarify that for me?
Yeah, no, that's perfect. Actually, I didn't mention it, so I'm glad you asked. We sampled the teat end as the cows were coming into the parlor, so before any prep was done. So this was on the teat end as they're walking in. The leap of faith says that if there was a bunch of strep species on their teat end, maybe that cow is more likely to get mastitis if it ends up inside the teat. We didn't report it because the numbers were too small, but we did do some, before and after prep and so forth to look at how well the prep was going. And that's obviously very farm specific and variable and sometimes even milker specific. So at this point, we weren't trying to get to that level. We were just trying to say, okay, is there a lot in the bedding and is there a lot on the teat ends? And we didn't really see a direct relationship that said a lot in the bedding, a lot in the teat ends. It was kind of like we see a lot of very low counts, and we saw that distribution of very few cows with high counts on their teat ends and more cows with low counts, which is a good thing, right? So we're still working on that pathway, but there's going to be a few little leaps of faith because this is kind of a population level study, not an individual cow where we follow that specific cow, all the way through. We're looking more at a population level. But we did swab them before anything was done, so we were trying to say what's happening in the bedding, what's happening on the teat end. Leap of faith, is this what's happening inside the udder of the cow and then ending up in the bulk tank.
This is Kathy Barrett again, and on behalf of the ProDairy program and dairy business, I'd like to thank you for taking the time to view this webinar.